Hello everyone, very good evening students. Good evening guys, welcome back. I welcome you all to this Biotecnica's YouTube channel. So today we are going to discuss NEET PYQs of biology. So today it is 7th day of NEET PYQ's Prerna series. So if you are new to this series and if you want to know more about the series, you can just write a mail to us at info@biotechnica.org. You can also join our Telegram group. The link you can find in the description given below. You can also call us on a toll-free number, the number that is displayed over the screen. So here I will be waiting for 1 or 2 minutes and then I'll start with the lecture. So welcome back everyone. Hi Vinod, very good evening. So you are having exam on 17th of July. So it's only 5 to 6 days that are left for your examination. So exam preparation is going all well. So come here and we will solve more numbers of PYQs, whether it is related to biology or chemistry. So here we are going to discuss mixed PYQs of both botany as well as zoology. Hello dear tech. Hi Mathi, how are you? Hello Sneha, very good evening students. So I hope I should start with the lecture. So the very first question you have on the screen is, the given figure shows a diagrammatic sketch of a portion of the human male reproductive system. So this is a diagram of human male reproductive system where you have to select the correct combination and you have to pick the correct option A, B, C and D from the given options below. The options are A, B, C and D. So I'll give you 30 seconds for this question. Just make it out and make sure the options that are given A, B, C, D, it's not in the correct order. Like it's not going from down. You have to see the picture and you have to answer accordingly. Yeah, come on guys, please answer for this question. What is the correct combination for this human male reproductive system? Option A, dear tech. Yes, you are correct. Because we know that this is the long muscular tube which is called as vasa difference. What is the function of vasa difference? It will take all the sperms that is there made in the epididymis. So it will take the sperms and it will send to the seminal vesicle. So the very first option would be vasa difference. You will find vasa difference very first and you can see it is only there in the two options. So option A and option B. So C and D will be eliminated here. So now let's see, it will take this sperm to the seminal vesicle. Now what is the function of seminal vesicle? It will add fluid to the sperm and this sperm will now be called as semen. So the second diagram or second feature, it should be seminal vesicle. As you can see the pink colored structure, it is seminal vesicle and it will take the sperm to the prostate gland. What is the prostate gland? This prostate gland will provide protection and nourishment to the sperm. So the C part would be the prostate gland and ultimately to the Cowper's gland, that is what called as bulbourethral gland. Now what is the function of this gland? This will add alkaline, it will provide the or maintain the pH of the sperm. So when the sperm enters the female body, it will neutralize the acidity of the urine and all. So the last part or D part, it is bulbourethral gland. So A is vasa difference, B is seminal vesicle, C is prostate and D is Cowper's gland. So the first answer is the right option here. So this is an diagram of NCERT book. You can see here, this is a structure of human male reproductive system. Now going to question number 2. So the fruit fly has 8 chromosomes that is diploid in each cell. During interphase of mitosis, if the number of chromosomes at G1 phase is 8, what would be the number of chromosomes after S phase? So here you have to provide the number of chromosomes that would be after S phase, after DNA synthesis phase. So what is the correct answer for this? Option A is 8, option B is 16, option 3 is 4 and option 4 is 32. Yes, please answer for this question. What would be the number of chromosomes at the S phase of the cell cycle? So here you are given G1 phase ka chromosome number. So G1 phase, it is already given 8. What would be during the S phase of the cell cycle? 16. Option B, DATEC. Now let's see. Now we all know that there are 4 phases of cell cycle. That is G1, S, G2 and M. Now this mitotic phase again consists of prophase, metaphase, anaphase as well as telophase. Now the chromosome number would remain constant or same during G1, S, G2, prophase as well as metaphase. When it becomes double when it comes to anaphase. Now in the question it is given about S phase or after S phase. What is after S phase? It is G2 phase. Now you can see the number of chromosomes would be same what is there in the G1 phase. Now in G1 phase it is given 8. So what would be the number of chromosomes in G2? Again it would be 8 because chromosome number they will change when it comes to anaphase of M phase. Now we'll see with the help of this diagram. I have put this tabulation that is for the human chromosome. Now in humans we are having 46 chromosome. In humans we are having 46. So you can see here this 46 number it is maintained. It is maintained till metaphase. When it is becoming double, it is doubling in anaphase. Anaphase and telophase, and again when daughter cells are made, so again this 92 will become 46 in 2. So it is becoming 92 or double in case of anaphase and telophase. But here we have to answer according to the question. So the right answer is 8. So the option A would be the right answer here. So now let's move to question 3. For assimilation of one carbon dioxide molecule, what would be the energy required in the form of ATP and NADPH2S? So this question is from photosynthesis. Calvin cycle. Yes, Jayatech, it is option A. So what would be the number of ATP and NADPH2 required in case of Calvin cycle? That is C3 cycle. So ATP is the energy source and NADPH2 is the reducing power. So what is the number of ATPs? So the options are, I will write the correct option for this, 2 ATP and 3 NADPH, 3 ATP and 2 NADPH, 5 ATP and 4 NADPH, 4 ATP and 5 NADPH respectively. So the first is for ATP and the second one is for NADPH2. So what is the correct option? So the correct option is 2 ATP, 3 ATP as well as 2 NADPH. Option B. How? Now we'll see with the help of diagram. Because we know in Calvin cycle, this ribulose by 1,5-bisphosphate, it will take carbon dioxide from the surrounding and it will fix carbon dioxide. Now this is 5-carbon molecule. When it gets associated with 1 carbon, it will form 6 carbon molecule. But how? 2 molecules of 3-phosphoglyceric acid will be formed. So this is a 3-carbon molecule. So 2 molecules will be formed that will compensate 6 carbons total. So here, now it will be converted to ribulose. This glycerate, how it is formed? It is formed with the help of ATP. So because this is 2 molecules of 3-phosphoglycerate, so 2 ATP will be consumed here. Now when this triose phosphate is formed during the reduction phase, now comes regeneration. This regeneration again will utilize 1 ATP. So how many ATPs total? 3 ATPs. And where this NADPH2 is utilized, it is utilized during the reduction process because 2 molecule of 3-PGA is there. So it will be reduced to triose phosphate or phosphoglyceraldehyde. So 2 ATPs, 2 NADPH will be utilized and 3 ATPs. So the right answer for this question is 3 ATPs as well as 2 NADPH for fixing 1 carbon of glucose. But we have to fix, if we have to fix pura glucose molecule, so glucose, it is having 6 carbons. So how many number of total ATPs would be there? 6 into 3, that is to fix 6 carbon of glucose. So 18 ATP as well as 12 NADPH. Why 12? Because 2 into 6. From where this 6 is coming? Glucose has 6 carbon. So the right answer according to this question is, because it is asked for 1 CO2 molecule or 1 carbon of glucose, that is why it is 3 ATP and 2 NADH. So here it is a match the following question. You have to match column 1 with column 2. So in column 1 it is given Bt cotton, adenosine deaminase deficiency, RNAi and PCR. So the correct combination is given option A, option B, option C as well as option D. What is the right answer for this question? Question number 4. If you find any one option correct, that option goes right. So here you can match with option 1 itself, Bt. We can see capital B and small t. So it is Bacillus thuringiensis. It is a name of bacteria. So B and T, it is matching with option 1 is matching with option 4. So where we can find A with 4, we can only find A with 4 in one option. So now we will check the further option or this option is the right answer. So now we'll further match if that options are correct or not. So PCR, we know we can quantify or we can detect any kind of infection in PCR. So where you can find the infection option, it is there in the third option, that is detection of HIV infection. You can do it with the help of PCR. So option D with third, you can find it in the first option itself. So the right answer is option first. What is RNAi? RNAi is a phenomena that is called as RNA interference that will provide cellular defense to the cell. What is adenosine deaminase deficiency? This is an enzyme, adenosine deaminase ACE. So this is an enzyme which is causing or which is deficient in the body and there lacks a B and T cell formation. So once we are providing this enzyme from outside, this is what called as gene therapy. We are treating the individual with the help of outside gene. So the right answer for B is first. So we can see B with first, C with third in only first options. So the right answer is option first. Now let's move to question number 5. In a population of 1,000 individuals, 360 belongs to genotype AA, 480 to AA, and the remaining 160 to small a. Based on this data, the frequency of allele A in the population. Again, guys, this question is from Hardy-Weinberg equilibrium. So here you have to find the allele for the dominant allele A. Frequency of the dominant allele, that is capital A. Is it option A, B, C, and D? What is the right answer for this? Yes, Mati and Daretech, you were correct. What is the right answer for this question? Now we know that Hardy-Weinberg equilibrium, P square plus 2PQ plus Q square is always 1. 5A. All right. Correct, Mati. Very good. Now let's solve this question. So it is given 1,000 individuals total, but for A allele, the frequency is 360. The frequency is for A is 360. That means P square, it is given 360. And 2PQ, it is given heterozygous, it is 480. And for recessive, it is given 160. So how to solve this kind of questions? So you are being asked about the allele in the question, that too of the dominant allele. So how to calculate the allele frequency? First of all, this is P square, that means the dominant allele. And well, elsewhere where you can find the dominant allele, you can see in the heterozygous. Now in heterozygous, we have A and small a. What is this A and small a? Whatever frequency is given, that means it is given 480. You have to divide it by 2. Why 2? Because you can see contribution of both the dominant and recessive allele in case of heterozygous. So when you will divide it by 2, you will get 240. Now this 240, it is coming from capital A also and 240 from small a. So now what we'll do? We will plus or add this 360 with 240. Now this is the actual contribution of the dominant allele, that is from the homozygous condition, and half A from the heterozygous combination. So what we'll get? We'll get 600. Now it is asking for dominant allele, that is allele A. Now the total individual that they have given in the question is 1000. Now we are having dominant allele contribution 600. So just we will divide 600 upon 1000, that will be 0.6. Cancelling 2 zeros, 6 by 10 is 0.6. So now for dominant allele, we are having 0.6 frequency. So this is only the question that is given. We have to calculate the frequency of allele A. So the right answer is option 1. And suppose if it is asking about recessive allele, so what would be the formula? For formula, recessive allele Q is 1 minus P. That means 1 minus 0.6 is 0.4. So the recessive allele के लिए अगर पूछा होता तो the right answer would be 0.4. But here according to question we have to answer. So the right is first option, that is 0.6. So now let's move to question number 6. Very easy question. Hi Mitty. Purines found both in DNA and RNA. Yes, purines are present both in DNA as well as RNA. So which is the correct option? What all purines that are present in both DNA as well as RNA? Is it option A, option B, option C, as well as option D? So you have to select the correct combination of purine that is present both in DNA as well as RNA. Option 2. Yes. Right, Sneha. Very good. Daretek. Mathi. Very good. Mithi. Now we'll solve how. Now we know that DNA structure, whether it is DNA or it is RNA, they both are having purines and pyrimidines. And what is purine? Purine bases are adenine and guanine. What is pyrimidine basis? It is cytosine and thymine in case of DNA. But in case of RNA, this thymine is replaced with uracil. But here we only have to answer about adenine and guanine, that is purine. So it is asking for purine basis, and you can see the purine basis, they are similar in both DNA as well as RNA. So the right answer is adenine and guanine. Here you can see thymine, but thymine, as I've already mentioned, thymine is not there in RNA. So it is uracil. So this option goes wrong. Thymine ka, this also goes wrong. Why cytosine is incorrect? Because it is asking for purine, but cytosine, it is a kind of pyrimidine. We have to answer purine for both DNA and RNA. So the right answer is option 2. Now question number 7. What is the colostrum, the yellowish fluid that is secreted by mother during the initial days of lactation, is a very essential to impart immunity to the newborn infants because it contains what kind of antibodies it is present in, or colostrum or yellow milk of mother? What kind of antibody it is present, or what cells are there in colostrum? So what is a colostrum? It is the yellow milk that is produced after pregnancy or during the lactation period. 7th D. Very good. Dare tag. Mitty, very good. The right answer is 7. 7 ka right answer is D. How? Because we know that colostrum, it is the yellowish fluid or yellow milk that is secreted by the mother during the initial days of lactation. And this colostrum, because it is a secretion, it is secreted from mothers. So it will always contain antibody. But what kind of antibody is present in the mother's milk? It is immunoglobulin A, because we are knowing that immunoglobulin A is the only antibody that is present in the secretion. Whether it is mucus, it is tears, it is milk, immunoglobulin A would be present. So the right answer, only one option you can find immunoglobulin, that is antibody. So the right answer for this question is option 4. Now let's move to question number 8. Again, match the following question. This is from sexually transmitted diseases. So you have to match column first with column 2 or list 1 with 2. So you have to choose the correct combination from the options given below. That is option A, B, C, as well as D. So what would be the right answer for this question? Question number 8. So the options are walls, IUDs, intrauterine devices, vasectomy, as well as tubectomy. What would be the right answer? Okay, some are going with option D, Mathi, Mithi, option 1. Okay, now let's see what is the right answer. Name itself suggests vasectomy. That means it is related to vasa difference. Where you will find vasa difference? In the option second. So where you can see C with second, you can see C with second only in one option, that is option 2. So you can clearly omit other options. So now we'll check other options if that is satisfied or not. Tubectomy. Tubectomy means it is related to female reproductive system. What? Fallopian tube. So we'll match D with 4. Yes, it is removal of fallopian tube. So the right answer for this question is option second, that is A first ka 2 ka first, B ka second, C ka second, and D ka fourth. Now, what for this valves used for? It will entry of sperm through the cervix is blocked. When the sperm is entered into the female body, but it is blocking the entry of sperms into the female body. This valves. What is this IUDs used for? That is intrauterine devices. It will cause the phagocytosis of sperm when it will enter within the uterus. And vasectomy, it is a removal of vasa difference, and tubectomy, it is for female reproductive system, that is the removal of fallopian tube. So now coming to question number 9. Conversion of glucose to glucose 6-phosphate, the first irreversible reaction of glycolysis, it is catalyzed by which enzyme? Here you have to select the name of the enzyme. Which is the correct option? First is aldolase, second is hexokinase, third is enolase, and fourth is phosphofructokinase. What is the correct option for this question? It is the very first step of glycolysis. Which of the following is involved in the conversion of this glucose to glucose 6-phosphate? Very good. Diatec, it is option D. Anyone else? It is the very first step of glycolysis. Now, what is the name of the enzyme that is involved in very first step? So now let's see. This is a glycolysis. You have to answer the very first step of glycolysis. You can see in the first step of glycolysis, this glycolysis, splitting of glucose. So this glucose, it is splitted into or converted to glucose 6-phosphate. So here, why 6-phosphate? Because one phosphate group is added. So who is adding this phosphate group? Kinase enzyme. But because this glucose is an hexo sugar, so enzyme name is hexokinase. So it will utilize, or it will take one phosphate from ATP and it will convert this ATP to ADP. And meanwhile, this glucose will be converted to glucose 6-phosphate. That means at 6th position, one phosphate group will be added, and that's why it is called as glucose 6-phosphate. So it is asking about the very first step. So it will be option 2, that is hexokinase. So now let's, and this, where this fructokinase is used, this fructokinase is used for the conversion of this fructose 2,6-phosphate to fructose 1,6-bisphosphate, but not from glucose to glucose 6-phosphate. So I'm sure this is clear to everyone. So now let's coming to question number 10. Now you have to select the correct matching in the following pairs. Here 4 options are given. You have to select the correct option. So which option is correct? Is it option A, option B, option C, or option D? Ninth, 10th question you have to answer. Select the correct matching in the following pairs. So now we know that there are 2 kinds of ER. It is smooth ER as well as rough ER. Now rough ER is known for the synthesis of proteins. Why? Because it has ribosomes in it. But what about smooth ER? It is known for the synthesis of phospholipid as well as synthesis of simply lipids. So where you can see this option, synthesis of phospholipid or lipid, you can find this option only in one option, that is option number B. Because smooth ER, it is given A combination के साथ oxidation of phospholipid. This oxidation of phospholipid, it is taking place in the cytoplasm but not in the smooth ER. If it is given synthesis of phospholipid, then this option is right. But here it is given oxidation. So this goes wrong. And about rough ER, synthesis of glycogen. No, synthesis of protein will take place. Glycogen synthesis will not take place in rough ER. So here in option D, rough ER, oxidation of fatty acid. Again, it will take place in the cytoplasm. Now only the one option is left, that is option B. So smooth ER, it will cause the synthesis of lipids or phospholipids. So the right answer for this question is option B. Because the function of a smooth ER that has no ribosomes over the membrane is the synthesis of phospholipid or simply lipids. Now here you have to solve this pedigree and tell me the answer. Here you have to calculate or solve this pedigree for this parent. You have to tell the genotype of parent A and B. So what would be the genotype of parent A or B? So I'll give you 20 seconds to solve this question because this is a pedigree. So take your time and write your answer. What would be the correct option for this parent? Here you have to tell the genotype of parent A as well as parent B. And make sure that in question it is asking about autosomal recessive. Now you know recessive condition, कोई भी disease recessive होगा, it will always express in recessive. जब वो उसके दोनों genotype क्या होंगे, small a or small bb. So what would be the right answer for this question? Now let's solve pedigree. Option A. Okay, all right. Very good, Datek. Now let's check if this is the right answer. So recessive means it will always show small a genotype. Now shall we write small a to this? Because this is recessive. This is infected. Now this small a, it is coming, this is a child of these both parents. So small a would be present here and small a would be present here. All right. But because this is infected, again double small a. Double A genotype would be there because this is infected, and it should be capital A and small a because this is not infected. But why small a I have mentioned? Because one allele, it is coming from this parent. Now again we'll go to this. This is again a small a, small a. Here we have to answer about this parent, parent A and parent B. This is infected. Why we are writing small a and small a? Because this is recessive. It will always contain double small a. But you can see when this pedigree is going in the next generation, this parent is also responsible for transferring this disease to this. So what we can say, whether it can be double A or it can be double A, capital A and small a. Why? Because this is not infected. You can't see any infection here. So it can't be small a, small a. It can be double A, capital A, or capital A, small a. Now let's match with the option. You will always see the B parent, it should be small a because this is infected. So where we can find a small a combination, it is only in the first option because this is infected and this is recessive condition. So here the genotype of B parent, it would be small a, small a. So the right answer is option 1. If it is given capital A for this A parent or capital A, small a, both are correct. Why small a is correct? Because somehow it is transferring disease to the next generation, as you can see here. But for this B parent, it is infected. So it would be small a, small a. So the right answer is this combination. Or this can be capital A, or capital A, small a for parent A. So I'm sure this is clear to everyone. No problem, Mitty. Now I hope this is clear to you. If you have any doubt, you may ask. Now let's move to question number 12. So it is again you have to incorrectly or select the incorrect match pair from the following. The first is chondrocyte, that is with smooth muscle cells. Second is neuron, that is nerve cells. Third is fibroblast, that is areolar tissue. And fourth is osteocyte, that is bone cell. Make sure you have to select the incorrect option, not the correct. So you have to select option A, option B, option C, or option D. What is the right answer for this question? Incorrect match pair. So what are the building blocks for these bone cells? What are these bone cells made up of? They are made up of osteocyte. So this option is correct. Neurons, they will make all the nerve tissue. Again, this is correct. Fibroblasts, they will constitute the areolar tissue. This again is correct. Then what about option A? Chondrocytes. These sites mean cell. Chondro matlab cartilage. The cell which will make the cartilage. But here you can see in the option that is not correct. It is smooth muscle cells. Here we don't have to talk about muscles because here it is given chondro. Chondro means cartilage. That means the option A is only the incorrect pair. Yes, very good, Mathi, Dertek, very good. So the right answer is option 1 because we have to select the incorrectly matched pair. Osteo means bone. The bone cells means osteocytes. Fibroblast, skin tissue. Neurons, they will make the nervous tissue or nerve cells. And chondrocyte, they will make cartilage but not smooth muscle cells. So the right answer is option 1. Now let's coming to question number 13. So during biological nitrogen fixation, we are knowing that we are having nitrogenase complex. So inactivation of nitrogenase by oxygen poisoning is prevented by. How this inactivation of nitrogenase is prevented? Is it by carotene or leg hemoglobin or xanthophyll or cryptochrome? Who will protect this nitrogenase complex from oxidation? This is question number 13. So how this nitrogenase complex is prevented? It is prevented by leg hemoglobin because we are knowing that this is the root nodules you can see, and inside this rhizobium will fix all the nitrogen from the atmosphere and give it to the plant. That plant will be rich in nitrogen, and that's why when we are consuming that plant ka part, we are getting high amount of protein. So what is this pink nodules? So pink nodule has leg hemoglobin, and because of this only the nodule color is pink. So what is the function of this leg hemoglobin? It will scavenge or it will remove all the oxygen that is there in the nitrogenase complex. Why? Because nitrogenase complex, it is very sensitive to oxygen. So when it will get oxygen, it will stop working. It will not fix any nitrogen from the atmosphere because it always needs reducing environment, not oxidizing. Oxygen free environment. So who will remove all this oxygen inside the root nodules? Leg hemoglobin. It will remove all the oxygen from this environment. So the right answer for this question is option 2, that is leg hemoglobin, and this is pink in color. So the rhizobium bacteria that is present in the root nodule of leguminous plant, it will fix the atmospheric nitrogen, yes, by an oxygen labile enzyme nitrogenase. Oxygen labile means oxygen sensitive. This is very sensitive to oxygen. So as to remove this oxygen, this leg hemoglobin is present. So the root nodules contain a pigment called red leg hemoglobin. Why pigment? Because this is pink in color, which prevents the exposure of nitrogenase to oxygen. So the correct answer is leg hemoglobin. So now coming to question number 14. Which of the following is the correct sequence of the event during mitosis? Again, we are having question from the cell cycle. You have to correctly select the combination of a statement or line by line statement what is happening in case of cell cycle or different stages of cell cycle. Is it option A, option B, option C, or option D? You have to select the correct statement that is taking place during mitosis. And make sure when I'm talking about mitosis, I'm focusing on M phase. So here you have to do according or solve this question according M phase ka steps. Very good, Mathi. Yes. Welcome, Prajapati. Now let's solve question number 14, where you have to select the correct sequence of events during M phase of cell cycle, that is mitotic phase. So now we all know that we are having 4 phases of cell cycle, that is G1, S, G2, M. And under M phase, we are having prophase, metaphase, anaphase, and telophase. So what happens in this stage? So firstly, the chromosome condensation will be there. So condensation will be there. So where you will find condensation? Option A, B, D and C and D. So all four option condensation is there. And then occurs the nuclear membrane disassembly. Nuclear membrane, it will break off. Why it will break off? Because it has to divide. Chromosome number has to be double. DNA has to be double. So what it will do? It will break the nuclear membrane. So disassembly of nuclear envelope will take place. So where you can find? You can find nuclear disassembly in option A as well as option D. So this option 2 and 3, it will go wrong. So now we are left with two options only. Now what will happen? Now once this nuclear disassembly has taken place, chromosome that is there, it will arrange to the equator. It will arrange to the equator during metaphase. So prophase, condensation has taken place, nuclear ka membrane ka all cleaving has done. Now it's a time for the metaphase. During the metaphase, these chromosomes are arranged at the equator. And after this equator, it will segregate and reach to the opposite poles during anaphase. And last comes telophase. Now we'll match the option. First is condensation. Here also is condensation, is option A. Nuclear membrane disassembly, and here also it is nuclear membrane disassembly. Yes. Now crossing over. When we are studying crossing over, this crossing over, it will come in meiosis, not mitosis. So this option goes wrong. We have smartly omitted all the three options. So the right answer for this question is option 4. Very good, Mati. So the right answer is option 4. Why? Because condensation will take place, all the chromosome will be condensed, then nuclear membrane disassembly will occur, arrangement of chromosomes towards the equator during metaphase, centromere ka division will happen, segregation of chromosome towards the opposite pole during anaphase, and ultimately telophase will come. That is PMAT. And here we have to answer only according to the question, that is mitotic, and this is PMAT. So the right answer is option 4. So here you can see the strategy how to deal with question. So always the one word question that are always tricky to answer. If you are knowing the correct answer, only then you can answer. But for this kind of statement based question on option correction, if you are knowing the sequence, only 3 or 4 sequence or 2 sequence, you can just omit the 2 option and you will get 50-50 kind of lifeline kind of thing. You will be left with 2 option only, and then you will go with the leftover options. So now we'll move to question number 15. Virids differ from viruses in having. How does virids and viruses differ? Is it option A, option B, option C, or option D? What is the difference between virids and viruses? How do they differ? Very good, Mithi. Very good, Mathi. They attack. So this is option C. Why C? Because we know that what is the difference between virus and virus. Now we'll draw. Suppose this is a virus. So virus will always have an outer covering or envelope. And inside this, the genetic material will be there. This is what called as viruses. What about viroids? It will always have single-stranded RNA, or you can just make it like this. It doesn't have this covering that you can see in virus. Then what about prion? Prion will always have single covering but not the genetic material. So this is the basic difference between viruses, virions and prions. So viruses, both the covering and genetic material would be present. For virions, only genetic material but not the outer covering. And what genetic material? It is always single-stranded RNA molecule, not DNA. So where we can see DNA, it is omitted because it is asking for virions. Here also we can omit DNA because in virions it is present single-stranded RNA. That too without a protein code. So RNA without a protein code. Because if I'm talking about viruses, so it will have a protein code, but not in virions. Virions only genetic material would be there. So the right answer for this question is option C. Very good, Datek. Now let's move to question number 16. The term polydelphous is related to. Is it related to gynoecium, androecium, corolla, or calyx? This polydelphous term, it is related to delphus. To what this term is related to? Is it related to pistil or gynoecium, androecium, corolla, or calyx? So this is related to androecium. So what is polydelphous? So in anther or androecium, what we are having? We are having anther and filament. So this is what the structure of one androecium, anther and filament. So if the filaments of androecium are joined to form more than two groups, but their anthers separate. When more of the filament will be joined together, but their anthers are separated, this is what called as polydelphous condition. It is called as polydelphous. Where you can see this condition? You can see this condition in citrus fruits. So this is from the NCERT line. When the filaments of androecium, they are joined together, but their anther is free. So this is what called as polydelphous condition. So this is a structure of one androecium when the filaments are joined, but the anthers are separated from each other. So this is what called as polydelphous. So this is for androecium. So now let's move to question number 17. Here you have to select the wrong statement. 4 statements are given. So here you have to select which of the statement is the wrong statement. Which of the statement is a wrong statement? Is it option A, option B, option C, or option D? So here you have to recall the work done by all the scientists, that is M.W. Beijering or mosaic disease, where it was done, Ivanovsky or W.M. Stanley. So here you have to recall all the concepts that you have learned in the previous chapter of class 11th NCERT textbook. Option 3. For 17, it is 3. Yes. So the right answer or the incorrect statement over here is viroids. Because viroids, it is an infectious particle that is containing a single-stranded RNA. This was discovered by Diener. This was discovered by Diener, not Ivanovsky. Ivanovsky has just talked about tobacco mosaic virus, and that too from a plant that was infected by this virus. So Ivanovsky has done this work, but viroids, it was discovered by Theo Diener. So the wrong answer or the correct option according to this question is option C. So viroids are infectious RNA particle, that is single-stranded RNA, which were discovered by Theo Diener when 1971. So these are devoid of protein code. Yes, we have already seen in viroids no protein code is there, only genetic material, and it will cause diseases in plants only. That means example of this is potato spindle tuber, chrysanthemum, stunt, etc. So now let's move to question number 18. Now here again this is from genetics. Gray is dominant over black. Which of the following would most probably get 50% black and 50% gray offspring? So here you have to select the genotype. What is the correct option? What would the correct option by which you can get 50% black or 50% gray offspring? Is it option A, option B, option C, or option D? Very good, Mathi. So what would be the correct option for question number 18? Make sure you have to cross this and you have to see how you will get 50% black and 50% gray. But this dominant is gray only and black is recessive here. 18B. So now let's see how. You already know about the test cross. What is this test cross about? When a dominant parent or a recessive parent, they are crossed, what will get in F1 generation? Will get small a or capital A ka genotype in F1 generation. When we'll cross this F1 generation to one of the recessive character, that is what called as test cross. And in test cross, the ratio would always be 1:1. As you can make out in the question, it is 50% black and 50% gray. When we divide, we'll get 1. That means the ratio is 1:1. So here you can just make it out, this is a test cross. Then what is the genotype for the test cross? It is the F1 progeny into recessive parent. So where you can find the heterozygous or F1 progeny, it is there in option 2 only. And again, GG parents. So what you will get, suppose if I'll cross this, if you want to cross check your answer, what I will get? I will get GG, again GG, small g, and small g. If you'll make Punnett square, you'll get 4 combination. And out of this is 50% dominant or gray, because you can see single G here. That means it is dominant. It would be gray in color. And the rest 2 will be black in color. That means 50% black and 50% dominance. So you don't have to do all this calculation because you are lacking time in case of competitive exam or whether it is NEET. So you have to be very smart while approaching 2 questions. So how you can do? You can just make it out. It is 50% black or gray. That means it is talking about 1:1 ka ratio. Ratio is always same. But when the ratio is same in case of genetics, it is same in test cross. टेस्ट क्रॉस का रेशियो कब सेम होगा? जब एक हेट्रोजाइगस होगा and one parent would be recessive. So you can find this type of option in option B only. So the right option is option B or option 2. So now question number 19. Which of the following statement is correct about recombinant protein formation? So this is from RDT or genetic engineering. So what is the right answer? Is it option A, B, C, and D? You have to recall the process of genetic engineering. Very good, Datak, Miti, Mati, very good all of you. What is the correct combination of the statement? That is during the recombinant protein formation. So what happens in genetic engineering? One DNA is taken, gene of interest, and one vector is taken. So what will happen? So in case of RDT, what happens? Firstly, we'll take the vector and we'll take the gene of interest. We'll digest both these things with the help of same restriction enzyme. Once this is digested, both will create the same sticky end. Now what will happen? They both will ligate together. Why? Because they both are digested with the help of same restriction enzyme. Make sure this restriction enzyme, it is same for both vector and gene of interest. Why so? Because you have to ligate this vector along with gene of interest. And once this restriction enzyme is same, only that time this sticky ends would be created same. So this will ligate with the help of ligase enzyme. Now you have to transform this vector or recombinant vector into the host cell. So how you will do this? You will just introduce this recombinant DNA or recombinant vector into the host cell. What kind of host it is? Homologous or heterologous host? Now we'll see. It is always heterologous host. Why? Because this gene that we are introducing from outside is not the gene that is present in the host. We are introducing it from outside. That means it is different. Different means hetero. Hetero means complex. So the host is always complex because this host, it is not having this gene that we are introducing it from outside. So the right answer for this question is option B. If any protein encoding gene in heterologous host, not the homologous host. And here, if you're not knowing anything about this question, how you have to approach to this question? Here, if you can find two options contradictory, you can see option A and option B, they are contradictory to each other. When you will find such a statement, only one option is like homologous or heterologous. When you will find such a statement, out of both, one option will always be correct. So if you are not knowing about anything, you can just omit other two options. Now you are left with two options. Still you are not knowing anything. Why? Because these both statements are contradictory statement. Out of these statements, one would be always correct. So now you have to recall what you have learned in the previous days. So it is always heterologous host. So now this option is correct. That is option 2. So now let's move to next question. Which of the following is not the function of antibodies? Again, antibody, which of the following is not the function of antibody? Is it agglutination, opsonization, this is neutralization, sorry. And the fourth is phagocytosis. Which of the following is not the function of antibodies? Not. You have to select or you have to see what is there in the question. Not is mentioned. Which of the following is the correct option? A, B, C, or D? Very good. It is phagocytosis. Why? Because we know that when antibody, they will combine with antigen, that will cause the agglutination of RBC or neutralization of this antigen. So this is the function of antibody, that is agglutination of this RBC ka particle or neutralization of antigen. Also, it does opsonization. What is opsonization? When it will enhance the phagocytosis by other immune cells. So this is one of the function of antibody. But phagocytosis, this is not the function of antibody. Why? Because phagocytosis, it is done by phagocytes. Now, what is phagocytes? You can see here the cell or the sites which will perform phagocytosis. Now, what are the example of this phagocyte cells? It is macrophages, it is NK cells, it is monocytes, etc., but not antibody. So the right answer, you have to select not the function of antibody. So if you are not knowing also what are the function of antibody, it is agglutination, opsonization, as well as neutralization, but not phagocytosis. This is performed by other immune cell, that is macrophages, NK, and monocytes. So the right answer is option 4. Now let's move to question number 21. Again, you have to select the correct sequence of gene that is there in the DNA fragment. So a DNA was cleaved using restriction endonuclease. So this is a DNA fragment. As you can see here, it was cleaved using DNA restriction endonuclease. So ABC produced as a digestion as shown below. Select the option that represents the correct electrophoresis separation of these fragments. So you have to select the correct option for this. Now 4 profile is given. You have to select what would be the correct digestion of this fragment. So how you have to approach to this question? You can just, I will give you a hint, you will just see the fragment size and answer accordingly. What would be the correct profile when these fragments would be running under electrophoresis? Welcome, Asati. Very good, Datak, Miti, Marti. Option B. Now let's check how to approach to this kind of question. Now it is given A, B, and C fragment. If you see the C fragment, it is larger in size. It is larger in size. What about B fragment? It is smaller. The most smallest one is B, and A is intermediate between both A and B and C. Both B and C, because B is the smallest, C is largest, and A is intermediate. So how to solve this question? It is asking for electrophoresis profile. So how to solve? We know that during electrophoresis, most the fragment will be smaller, farther it will move. It will move farther in case of electrophoresis. Why? Because this is smaller. So where you can see who will move faster? The one who is smaller. So who is smaller here? Only B is smaller here. So what fragment will get at last? Will get B. Why at last? Because B will move more distance. Why? Because this fragment is smaller. So where you can find B? You can find B in option 4 as well as option 2. So A and C are omitted. And you can see the option C or the C fragment, it is a larger fragment. When the fragment is larger or the DNA segment is larger, it will always retain in the agarose gel electrophoresis. It will not move. Why? Because it is caught in the pore size of agarose. So you will always see option A in the right answer. Option A means this fragment is bigger, C is bigger. So C will be here. It will be retained in the first and B will travel more distance and A will be intermediate. So the right answer is option 4, because smaller the fragment, more distance it will travel in case of agarose gel electrophoresis, and larger the fragment, it will retain near the voltage. It will retain near the negative terminal. Why negative? Because DNA is negatively charged. So we will load all the samples toward the negatively side. It will not move. Why? Because this is the larger fragment. So what is the electrophoresis profile? It is C, A, B. B is a smaller fragment, A is intermediate, so it will move to some distance, but the C you can see here in the question, it is the larger fragment that is given. So C will not move at all. So the right answer is option 4. So I'm sure this question is clear to everyone. If you are not clear with this question, you may post your queries here. So the right answer is option 4. So now let's move to, if this is clear, now let's move to question number 22nd. Now again, this is match the following question where you have to match column 1 with column 2nd. So here again the list of antibodies are given. Here you have to match the name of the antibody or the functions of these antibody. You have to select the correct combination out of these 4 combinations given below. Very good, Asati. All right, Mithi. Thank you, Asati. Now let's practice question number 22nd. We have 5 more minutes. So we'll practice this question. Last question. We have 4 more questions left. 22nd ka D. Now let's see. I've already mentioned in the previous question that A antibody, it is found in the secretion. So this antibody, Ig means immunoglobulin. Immunoglobulin means antibody. So here we are talking about immunoglobulin means antibody. But this antibody, it is found in secretion, whether it is mother's milk, it is tears, it is saliva, it is mucus, anything. So this will match with option 3. So the third option, it is with A with 3. Where you can find A with 3? Only in one option. So here you can see smartly how you can only by selecting one option only, you can clearly go through the right answer. So again, we'll check further options, what if it is correct or not. So IgD, this antibody, it is always present over the surface of B cell. Why B cell? Because we know B cell का जो plasma cell होता है, it will form antibody. So IgD is the antibody that will be presented over the surface of immune cells. So it will match to option 4. So B will match to 4. Yes, we can see B will match to 4. So it is the surface antibody that is present over the surface of B cell. Why we are talking about B cell? Because this antibody, it is secreted by B cell. Or B cell ka kaun sa form? Plasma cell. Because when B cell gets activated, it will be differentiated into plasma cell as well as memory cell. This plasma cell is the effector cell that will form the antibody. And once this antibody is formed, it will be presented over the surface. So what antibody it is presented? It is IgD. IgE, it is always formed when there is some kind of allergy in the body. That is why IgE is also called as allergic antibody. So we can see C with 2nd, and IgD is called a universal antibody because it is performing many functions in the immune host. So D will match with 1. So the right answer is option 4. So I'm sure this is clear to everyone. Now let's move to question number 23. If the total amount of cytosine and guanine in a double-stranded DNA is 60%, then what would be the amount of adenine in this DNA would be? Here you have to apply Chargaff's rule. What is Chargaff's rule? If you are knowing the Chargaff's rule, you will do this question. So is it option A, B, C, and D? What would be the amount of adenine in this DNA? 20%. Now let's see how to solve this kind of question. Because Chargaff rule, it is saying that the guanine, whatever guanine it is present, what are the examples of guanine? It is A and G. And for cytopurines, pyrimidines, it is cytosine and thymine. So how to apply this rule? Chargaff rule says the sum of guanine will always be equal to cytosine, because we know that guanine pairs with the cytosine. So the guanine will always be equal to cytosine. So here you can see guanine and cytosine, it is 60%. That means 30% would be guanine and 30% would be cytosine. So the leftover 40% would be for adenine and thymine. Why thymine? Because I'm talking about double-stranded DNA, not RNA. If I'm focusing on DNA, that means it would be thymine. So jitna bhi adenine hoga, utna thymine hoga. That means 20% adenine and 20% thymine. So the amount of adenine will in the DNA, it would be 20%. And if you have the question like you have to calculate the leftover amount of bases or adenine and thymine put together. So what would be the total percentage? That time it would be option 3. But here it is only asking about adenine, so it is option B. How we are solving this question? Because we know that guanine, it always pairs with cytosine. So jitna bhi cytosine would be there, the same amount of guanine would be there in the double stranded DNA, and the adenine jitna rahega, thymine would be in the same amount. So like this we are solving this question. So 60% it is always already given for cytosine and guanine, and the leftover is 40%. Why leftover 40%? Because we are considering 100%. कोई भी चीज 100% होगा, so 60% is already occupied. अब 40% किसे मिलेगा? Adenine and thymine के combination. But here we have to solve for adenine, so it is 20% divided by 2. So I'm sure this is clear to everyone. Is it? All right. So now let's move to question number 24. This is the last question from genetic engineering. That is, a cloning vector has 2 antibiotic resistant genes. A cloning vector has 2 antibiotic resistant genes, that is for tetracycline and ampicillin. A foreign DNA was inserted into the tetracycline gene. A foreign gene is inserted and this site is blocked. Non-recombinant would survive on the medium containing. Is it option A, B, C or D? How would non-recombinant would survive? They will survive in ampicillin or tetracycline or in both the plate or in none of the plate. What would be the right answer for this question number 24? Option B. Welcome Saloni, very good evening. Option B. So now let's see how to solve this kind of question. The right answer is option C. I will tell you how. So suppose here it is given for vector. This is AMP resistant. This is ampicillin marker. This is tetracycline. Now if I am inserting some kind of gene over this, so this would be disrupted. ठीक है? और क्या होगा? Antibiotic resistant would be made. Ampicillin is intact. It is not disturbed, but here we are having new gene of insert, that is foreign DNA. So this site is disrupted. So what will happen? If I will transform this it in a host, what I will get? I will get कि now colonies, they will not grow in tetracycline. Why? Because the vector is not having tetracycline resistance. How? Because this tetracycline, it is already disrupted by some kind of foreign gene. Why some kind of foreign gene? Because we are making a cut in the vector and we are inserting this foreign gene. That means tetracycline is not there. But the question is not asking for recombinant. Question is for non-recombinant. Non-recombinant means nothing is inserted. If nothing is inserted, then the vector would be circularized or intact rahega vector mein. No disruption has happened. If no disruption has happened, that means ampicillin and tetracycline sites are intact. These sites are not disrupted. So they will grow both on AMP and tetracycline plates because this is not non-recombinant. Not recombinant. If I'm talking about recombinant, that means I'm making a new combination. Vector with some foreign DNA. But here it is asking about non-recombinant. I'll go back to the question and I'll show you. This is asking about non-recombinant. Non-recombinant matlab gene has not inserted into the vector. If the gene has not come inside the vector, how this tetracycline will be disrupted? It will not get disrupted. Why I'm talking about tetracycline? Because question mein bola DNA was inserted into the tetracycline. But this is non-recombinant. So it will grow on both the plates, whether it is ampicillin or tetracycline. So the right answer is option C. But if it is asking about recombinant, that means it has lost its tetracycline resistance. That means the colonies that will grow, it will kill on tetracycline. That time it will only survive ampicillin but not tetracycline. But if the question is for recombinant, but here we have to answer for non-recombinant, so it will survive in both the place, whether it is tetracycline or ampicillin resistance marker. So the right answer is option 3. So I'm sure this is clear to everyone. If not, you may post your query. So the last question is, which is the characteristics of growth? How growth is defined? Is it increase in mass, increase in number? Third option is increase in ability to sense, or both 1 and 2. What is the right answer for this question? How growth is defined? Growth is defined by the increase in number and the mass. So growth, it is a characteristic of all living cell by which a cell will increase in size. It will increase in size or number. Size means mass. That means the right answer is option 1 and 2 both, because a cell increases in number and also size means mass. So the number and mass will be increased. That means option D. So the right answer is option D. So here we have practiced 25 questions, and that too from botany and zoology. And more, we have focused on numericals and strategic questions, how to deal with different kinds of questions, whether it is match the following question, numerical, genetics problem, inheritance, all things we have dealt. So now, thank you everyone. This was last session from my side. Tomorrow you will have more sessions for chemistry and biology, and then you will write your NEET exam. So thank you everyone for watching this session. If you like the session, do not forget to like, share and subscribe. If you have any queries pertaining to this lecture, you just post your queries in the comments box or in the description below. You can see I will give your answers or query. I will solve your query, whatever it is regarding to today's lecture. So thank you everyone for joining and for your time and patience. Meet you in the next video. Till then, bye bye, take care, and wish you all the best for your NEET examination, because every one of you is going to write the examination, and I'm sure you will definitely get a seat this time. So wish you all the best. Take care of yourself. Bye bye. All the best.